Alrighty, cellular respiration round two. Here we go. We just have the electron transport chain and a couple other things to clean up, so shouldn't be too bad. Be shorter than the other one anyway. So, as we talked about the other day, the point is to make ATP. So, how do we do that? I showed you this diagram early on during glycolysis, and the goal is this ATP synthase is going to set up a hydrogen gradient and allow the hydrogen to flow through that ATP synthase. And then we're going to generate some ATP that way. So we'll get there. We're not there yet. This is, again, what we're working for. And I'll show you more details on this once we get there. But this is what we're trying to get to so we can generate some ATP. Not there yet. Accounting so far: glycolysis 2 ATP, Krebs 2 ATP. It's going to take a lot more than 4 to run ourselves. So that stat down there in the yellow box is pretty crazy. A working muscle recycles over 10 million ATPs per second. So we need a lot of it, okay? And we have to go through a lot of ATP. So there has to be a better way. So the electron transport chain, a series of proteins built into that inner mitochondrial membrane called the cristae. And what's going to happen is there are going to be a series of reactions going through there, and we're going to then set up a hydrogen gradient, proton gradient, that's going to power that ATP synthase to make some ATP. So we get about 34 ATP from one glucose with the electron transport chain. So if you were expecting millions, I'm sorry to disappoint you. So overall, that's only about 38, right? Because we made 4 in the first couple steps. But keep in mind, that's only one glucose. How many glucose molecules do you think are in a Snickers bar, any candy bar? Probably millions. So, you take that times 34 and you start making some headway. I mean, cookies, pretty much all glucose, right? Tons of glucose in there. So you take that times 34 and you can start seeing how things add up quite a bit. So again, ETC is only going to happen in the presence of oxygen, aerobic respiration. So now we're getting somewhere. Here's where we're at on our respiration map, if you will. So been through glycolysis, been through Krebs over there in the mitochondrial matrix. Now we're going to be hanging out in the cristae, or the inner membrane of the mitochondria, for the electron transport chain. We're ready to crack open the piggy bank. We have all of our electron carriers ready to go, and we're going to do what's called oxidative phosphorylation. So now a different type of phosphorylation that we're going to get to down the road here, and we're going to get some oxygen involved in this one. So little mitochondria background, which most of you have seen before. Double membrane, you have an outer and an inner. The inner is really highly folded. And then you have the inner membrane space, which is fluid filled between the membranes, which is that matrix where the Krebs cycle takes place. So now we're going to blow it up. So just that one little circle there, that piece of the membrane, we're going to blow up and put it up here. So you can see that you have a lot of these protein complexes. Oh goodness, went too fast. It wasn't catching up, and then it did. You can see that you have a lot of these protein complexes. So all of these big blobs right here, these are your proteins that are lodged in between your membrane right here. You can see the phospholipid bilayer of the inner mitochondrial membrane right there. So you have NADH dehydrogenase, you have cytochrome BC complex, and you have cytochrome C oxidase complex. Not important for me. I won't ask you about those. Don't think college board will not ask you about those either. But doesn't mean you shouldn't know that they're kind of there, hanging out and doing their job. Okay, so mitochondrial matrix on the inside, the inner membrane space here, and we are in that membrane there. So that's kind of where everything is, and we'll fill in some gaps here as we go. So we generated a lot of electron carriers from glycolysis and Krebs, so we're going to have those ready to go. All the NADH and FADH, they are going to be good to go. So we got a lot of those guys. So time to break open the piggy bank. So here's what's going to happen. So when that NADH comes over to drop off that hydrogen, right? Think about a hydrogen. Hydrogen's one proton and one electron, and you can see it there in the picture. And so they're going to do kind of different jobs here. And so here's your NADH, and it's dropping off the hydrogen, and now it's NAD plus. And where do you think that NAD plus will go? This NAD right here. So this guy right here, what's going to happen to him now? He's just an NAD plus now. So what could he possibly do? Well, he can go back, right? He can go way back there to the beginning, and he can go back to the Krebs, and then kind of pick up some more protons, some more electrons to bring back to the electron transport chain. I like to refer to NADH and FADH. I told you earlier they were electron shuttles. It's kind of like the airport shuttle bus. So I'm sure a lot of you guys have flown on a plane and you park somewhere away from the airport and you hop on that little shuttle bus that they have to take you over to the airport. So a bunch of people get on from the parking lot and they take you over to the airport and they drop you off, and then it's empty now. Okay, it's empty. It dropped off its cargo, and now it goes back to the lot to get more, and then it goes again and back and again and back, and it just runs that loop. And that's kind of what this NAD plus is doing. He's just the shuttle bus. He's picking up the electron, bringing it over to the ETC, dropping it off, and then going back to get more. So that's kind of how that goes. So now we have our electron. Okay, so we dropped off the hydrogen. Here's our electron, and that electron is going to get it's going to power protons being pushed out into the inner membrane space. And then the electron is going to travel horizontally. It's going to go through the electron transport chain. Okay, it's going to go through all these different proteins, and it's going to, the energy of the electron is going to power the proton pumps. So we're going to pump hydrogen out into that inner membrane space, and then the electron is going to end up going through all of the complex there. And FAD, same thing. There's an FADH2 there now in the middle, and it's going to drop off its electrons and do the same thing. Okay, so it's going to power the hydrogens going out there. And so why would the electron want to go through there? What's really pulling it through there? Well, it's actually going to be oxygen. Oxygen is what's pulling that electron through there. And so when the protons come back through, we're going to end up making some water with that oxygen right there and the electrons that it's accepting. Okay, so we'll cycle through this a couple of times. I know this picture's okay, but it could be done a little bit better. So electron carriers are going to pass the electrons and the protons to the ETC. The hydrogen gets cleaved off. The electrons are stripped from the hydrogen atoms, and the protons are then pumped out into this inner membrane space right here. So now all this area out here is getting flooded with hydrogen. Okay, it's getting flooded with protons. And so what happens when you build up all this one type of molecule on one side of the membrane? What do those molecules want to do? Okay, so here we're passing out all these hydrogen over here. All these protons are going out to the inner membrane space. Well, what do they usually want to do if there's a big group of something? All things want to spread out, right? They want to go back across the membrane. So they are actually they get pumped out here. Okay, they get pumped out into this space, and here's this big group of hydrogens now. Well, what do they want to do? They want to go back through, and the ATP synthase, which is this big purple guy right here, this is the enzyme, the hydrogens are going to come back through here and ultimately go all the way through, and that is going to be, what turns that turbine right there. It actually looks kind of circular. If you look really close to right here, there's a black arrow there showing you that it's going to rotate in a circle. Okay, so we pump hydrogens out just to get them to come back through, but to come back through the ATP synthase. Okay, so what pulls those electrons down the ETC? I kind of already tipped you off on that one. So oxygen. Oxygen is the biggest electron acceptor in biology. It may not be true for chemistry, but as far as biological systems are concerned, oxygen, if you think about where oxygen is on the periodic table, what's its number? Okay, what's its number? Its number's 8. right? So there's 2 electrons in the first ring, and then there's 6. So what does it want? It wants electrons. It wants to accept electrons to get to be happy again, right? So you have, here, my pen, have the electrons zipping through here, going all the way down. You have the protons getting pumped out here. We're making this big hydrogen gradient out here. And then you have your protons coming back through this way, okay? And that's going to spin the ATP synthase, and I'll get back to that in a second. The oxygen is what's pulling the electrons down, okay? So the oxygen pulls things in, and it's going to accept the electrons, and ultimately that means it's going to make some water, because those hydrogens, you have some hydrogens flowing through here too, and they're going to join up with the water, and they're going to make some water molecules. So that's where it comes from in the equation. That water that's on that side of the equation, that's where that comes from. Okay, so the electrons will flow downhill all the way to oxygen, because each thing in the electron transport chain wants the electron just a little bit more, but oxygen really wants it. So it goes downhill till it lands on oxygen. Okay, and that is oxidative phosphorylation. Okay, so a little bit different than substrate. We're not grabbing a phosphate and sticking it on from a substrate. It's this process right here that's called oxidative phosphorylation that's driving the synthesis of the ATP. So here's your electrons flowing downhill, where each carrier is a little bit more electronegative than the other. It means it wants the electron a little bit more than the other. So it's kind of like bouncing a ball down the steps, where it just kind of keeps going down. Okay, in a very stepwise controlled reaction. Don't want to have any PEEP disasters going on. Okay, so just kind of a little visual there for how those electrons are flowing down through everything. So we did it. We finally set up the gradient. The protons are going to flow through that ATP synthase. It's going to spin, and it's very hard to see how this actually works in a still picture. So I will post a video on how this ATP synthase actually works. It's only about 3 minutes, but I hope you watch it. It'll make more sense to you once you see the video. The protons actually are responsible for turning that thing in a circle. And then that's the other aspects of it down there at the bottom. They're not stationary. They kind of move in and out like this. And as ADP and phosphates come into them, they are going to kind of squish them on there, for lack of a better term, and make some ATP from just ADP and phosphate. And we call that the proton motive force, because it's the protons that are making that thing go round. Chemiosmosis, another little vocab word for you here, proposed by Peter Mitchell, won the Nobel Prize for it. So it's the diffusion of ions across the membrane. Keep trying to get out of the way here. So the buildup of the proton gradient, just so hydrogen could flow through the ATP synthase enzyme to build ATP. So the idea that it pumps it out and gathers in a big enough group to where it wants to go back through from high to low, right? Doesn't cost energy for it to come back through. That idea that you pump it out just so it'll come back through, that's called chemiosmosis. It's basically osmosis of chemicals. Okay, if you want to break it down like that, it's coming back through there, through that process. Okay, and it links the electron transport chain to the ATP synthase, which is what Peter Mitchell there figured out. Pretty impressive. All right, so we got a little diagram here, a little summary diagram. So just follow the things as they pop up. I will tell you before we get going. Here's the pyruvate from the cytoplasm. So what process is not on here? Glycolysis. Okay, this is everything since glycolysis. So glycolysis happened out here in the cytoplasm, and we made a couple pyruvates. Okay, so it picks it up from pyruvate right here. And so here's the transition step, and I'll start clicking now so you can kind of see the things fill in as we go. The transition step, it comes in, makes some acetyl-CoA, right? It made a little NADH at the time, and it's going to enter that Krebs cycle, and it's going to spin around in the Krebs cycle, and we're going to make some FADH. We're going to give off some CO2. We're going to generate a little bit of ATP, and then it's going to keep going, right? And those electron carriers that we're generating are going to head over to the membrane, to the cristae, and the electrons are going to get harvested there where that number 1 is. And so they are going to start, the electrons are going to start flowing through the ETC, and it's going to create that proton pump, and it's going to push those protons out into the inner membrane space, and they are going to want to come back around and then come back through the ATP synthase. So protons get pushed out all the way out here into the space. Electrons are flying through here and they're energetic, so that's what's powering the protons getting shot out there. And then your hydrogens, because of the gradient now, they want to come back through the ATP synthase. So this is where we're going to generate most of our ATP. We have our oxygen, which is pulling the electron down. We have our oxygen here, and it's going to join with the protons to form water. Okay, so here's our water that we're generating. And protons will diffuse, like I said, back down the gradient, driving the synthesis of ATP. So there's kind of Krebs and the ETC together, and they seem like very separate things when you look at them on the slides, but they're right next to each other. They just move electrons very quickly. So it's not a very far distance at all. And there is, I didn't mention this earlier, but this is only one electron transport chain. You guys know that the cristae is all wound up in there and all kind of curly all up in there so that there's more surface area for reactions, right? So do you think this is the only electron transport chain happening inside of a mitochondria? Nope. Okay, each mitochondria has lots of electron transport chains happening simultaneously, and you have lots of mitochondria inside your trillions of cells. So now maybe you can see how the numbers of the ATP build up and get us what we need. So a little ETC summary. Inputs, 10 NADH, 2 FADH, and some oxygen. Outputs, per glucose, 32 to 34 ATP, 10 NAD, which will then be recycled, and 2 FAD+, which will then be recycled. Okay. So here's your picture again, your overall cellular respiration map, and you can follow the electrons and all the processes through there. We made 2 ATP there, substrate level phosphorylation. We made 2 ATP there, substrate level phosphorylation. And we made about 34 ATP there, oxidative phosphorylation. Okay, so that's a little summary for you. About 38 total per glucose. All right, so summary of respiration. Here is your formula of respiration. So where did the glucose come from? Comes from food, right? Comes from food we take in. Where did the O2 come from in the equation? Plants, ultimately, right? Plants gave it to us. We breathe it in. Where did the CO2 come from in the equation? We generated most of that during Krebs, right? During that transition reaction, we generated some CO2 there. In both the transition reaction and in Krebs, there was some CO2 that gets generated. Where did the CO2 go? Out. Breathe it out for the plants. Where did the H2O come from? The water. It was generated in the ETC right there at the very last step. Where the ATP come from? Well, all three, right? 2, 2, and 34. What else is produced that's not listed in the equation? NADH and FADH get produced all the time. They just get recycled back. Okay. Why do we breathe? Why do you actually bring in air? Most people just go, to live. Okay, well, it's a little more complicated than that now, right? Hopefully you can give a better answer to that question, is why you need oxygen in your body, in your cells. Why do you need that oxygen in there? Pretty important part that oxygen plays in that last step, right? It's the final electron acceptor, right? That's oxygen. That's what it does. So it drives that ETC, which is where we get most of our ATP from. So what happens if O2 is unavailable? The ETC backs up. The electrons don't land anywhere. Then the NADH doesn't drop them off anywhere. And so everything gets backed up. Nothing's pulling the electrons down the chain. They can't unload. ATP production ceases. Cells run out of energy. And then you die, right? So there's a lot of things that have to happen there. And it's all driven by oxygen, okay? So you guys probably heard of cyanide, right? You've probably seen cyanide in movies and TV shows. You know it's a poison. When people take cyanide, a lot of times it happens in spy movies where they put a pill in their back of their mouth. If they don't want to be caught because they have secrets and they want to take their own life, they'll put a cyanide pill in there so their secrets die with them. Well, cyanide actually affects cytochrome C, that last complex there, and it pokes holes in that membrane, and then it breaks down the electron transport chain. So same idea, right? Same process happens. No ETC, no ATP. Your cells die, and off you go. So what's the point? Made some ATP. Okay, glucose is important, but it's just the model. Okay, it's the one we focus on the most, but it's just the model. Different components can be put through this process at different points. If an animal is starving, it will degrade the molecules in the following order. So the carbs will go through first. Your body will rip through your carbs in your body first. Then your fats. It takes about 3 weeks to get through all of your fats. And then proteins only at the very end. If your body starts breaking down your proteins, you in trouble. Okay. So over here, this is the carbohydrate pathway, which is the pathway I just showed you all the way down. And then proteins, depending on the protein, they can enter at different points along this process. And the same with fats. They don't start at the same place, but we can still get some ATP from some of those molecules. It's just we don't focus on them very much. Glucose is just kind of the model. Carbs are the model, and that's how we want you to learn it. And then, if you go into grad school, you'll learn some more about these other things if you really want to get into it. We like to be able to control, right? In these cellular reactions, control is a good thing. Every step is controlled by enzymes. So there's always some control happening with these enzymes, as long as everything's working properly so they don't go out of control and don't cause us problems. Here's a fun one for you. Phosphofructokinase, okay, is one enzyme under significant study because it's the main enzyme or one of the main enzymes in glycolysis. And glycolysis is really well conserved, as you know, over all living things. So phosphofructokinase up there, look what happens. So AMP, if you have a lot of adenosine monophosphate, if you have a lot of AMP, that's going to stimulate phosphofructokinase to do its job and start kicking out and start running through. I should probably grab my pen. So if AMP stimulates phosphofructokinase, then this process is going to run and we're going to end up with some ATP, right? That will lower the amount of this. Okay, if we get more ATP, then we'll have less of that. So if we have a lot of citric acid right here, if we get a lot of citric acid in the Krebs cycle, well, too much is going to come up and it's going to inhibit, and it's going to stop this process from working. Because if you have a lot of citrate just sitting there and you're not needing the ATP being built, why would you do it? Why would you keep building ATP if you didn't need it? And the same thing over here, if you make a bunch of ATP, okay, if you make too much and you made a bunch and then you decide your body's not active and you're sleeping a lot or whatever, then it's going to inhibit this enzyme. ATP itself can inhibit phosphofructokinase from actually working. So that's how it kind of gets controlled. So where we make it when we need it, but we don't just make it when we don't need it. We don't want to be wasteful, right? We want to use the ATP appropriately. And there are some review questions for you. And now you are going to be experts on cellular respiration. See you later.